We're going to explore metagenomic analysis through the NMDC workflows today. We have data from the Spruce project that we have available to you and already run through the NMDC workflows. And we'll be discussing some of the important aspects of metagenomic analysis, and hope that that helps both novices and maybe some more experienced people as well. Our lead today, our plenary speaker, will be Marie Kroeger from Los Alamos, and she's going to tell us how she uses soil metagenomics in her own research.
Great, thanks, Karen. So I guess I will share my screen here. Wonderful. Well, thank you all for joining us today from wherever you're located virtually. And thanks to the organizers for inviting me to speak today. I'm super excited to be here to talk about metagenomics, which I view as a foundational tool for understanding your soils. So first off, just to give a bit of an overview, I'm going to talk about what soil metagenomics is for anyone who maybe isn't super familiar, and what it can and can't tell you about your data and your ecosystem. And then I'm going to share two different stories of how I've applied metagenomics in my research previously. And then I'll end today by just talking through a few different takeaways, things to consider if you plan on using metagenomics for your soil research. So let's get started. First, what is metagenomics? Metagenomics is when we extract the total community DNA from, in this case, our soils, and we analyze that using different sequencing technologies. And so this is a little bit different than maybe amplicon sequencing that you've heard of before, where it's focused on a specific gene. A lot of times we use a phylogenetic marker gene like 16S for bacteria and archaea, or ITS for fungi. Here, we're taking the total sample DNA, and it's going to be able to tell us who's present in the sample, what genes are there, and the genomic and functional diversity within and between our samples. But it's important to take into a consideration a few key points about the limitations of metagenomics. But before I get into that, I want to highlight what sort of the analysis steps are for metagenomics. And I know that others are going to go into a lot more detail. And if you're in the full summer school, you guys will get to play around with some data, which is exciting. But I want to highlight a few key steps that we have to do when we're analyzing our metagenomes. First, before we do anything, we have to do quality control. So this is when we're going to trim our reads based on quality scores, remove any artifacts that might be there, or any linkers or adapters that you've used. And sometimes we add a spike in, so you want to make sure you're removing that. Once we have our nice, clean, filtered reads, we can then do taxonomy classification. And I know that with the NMDC workflow, you guys will get to see how that works in more detail. After we've gotten our taxonomy, we then move on to do assemblies, which we can feed into annotation pipelines to do feature prediction and understand what genes are present, how they might be associated with, for example, different biogeochemical cycles. And then the last step is a more recent development in metagenome analysis, probably in the last 10 years, maybe a little less than that, where we've added a step where we look at metagenome assembled genomes. And this is when we're going to actually assemble population genomes using different tetranucleotide frequencies and relationship between our samples to identify microbe A versus microbe B genomes. So things to consider. One thing, which is sort of a pet peeve of mine, so sorry if I get on my soapbox for a second, but differences between DNA extraction kits. When you're doing your planning your experiments and going to take DNA from your soil samples, it's really important to think ahead and think about the fact that there are different biases associated with DNA extraction kits, and you can go into the literature and find a lot more detail about this, but it's important to try to plan ahead and make sure you're using the same kit across your different soil samples. The next point I want to make is that soils have extremely high species diversity, which probably many of you already know. On the right here, I found this figure from Jennifer Martini's lab, where they were showing the differences in richness between soils and blue. Orange is our sediment. So even just between soil and sediment, you see this decrease in richness. And another aspect is that in soils, there's also high beta diversity, so higher turnover between our soils, which leads to an overall really high diversity. And a lot of times this is important to understand because that means we need to do deeper sequencing. This is especially important if you're focused on a really specific function. So in my case, I'm going to be talking to you guys about methane-cycling. And if I don't have a deep enough metagenome, then I'm not even going to be able to do any sort of statistical analyses looking at my different genes associated with methane-cycling. So it's really important consideration. Another point to make is that there is what we call relic or environmental DNA a lot of times in soils. So this is DNA that is just in the soil, but not actually inside a microbe. It's just sort of necromass DNA sitting around. And so an important thing to think about when you're designing your experiments to use metagenomics is that alone, metagenomics does not tell us whether a microbe is alive, dormant, or dead, and whether or not it's expressing the genes that you're getting annotated in your metagenome. However, you can actually get to that aspect of whether a microbe is alive and expressing that genes if you combine metagenomics with some other techniques that I want to highlight for you. So first off, there's a really exciting new technique that maybe you've heard of called BONCAT. And hopefully I won't butcher this, but it's bioorthogonal, non-canonical amino acid tagging, but just call it BONCAT. And you can use this technique or other viability dyes to be able to identify the active fraction of your soils. So on the left here, I'm showing just a really simplified example where you added this amino acid analog to your soil. And if a microbe is active, it's able to take it up. And one of the nice things about this is that it doesn't rely on replication. It's just going to take up this amino acid, incorporate it into the protein. So it's a pretty nice technique to use. However, one of the challenges that maybe you're thinking about is like, it's hard to get cells out of soil, especially if you want them to stay intact and to do cell sorting with flow cytometry. So there have been successes, but this technique's been more applied to marine ecosystems and some sediments. I think it's going to grow in soils, but so far it's not been applied as often to soil systems. However, stable isotope probing has been applied to soil quite successfully. And this is when you use a stable isotope form such as 13C or 15N, 18O. Those are all really common. And you inject this labeled isotope into your soil sample, along with always having your control isotope as well. And basically, if a microbe is able to utilize that labeled substrate, so for example, if we were using 15N ammonia, if it was able to take up that nitrogen, it would then incorporate that into its DNA or RNA or proteins, and you can isolate that and understand what the active fraction is. So if you use this technique, you would, in this case, let's say we're doing DNA stable isotope probing, we would then take out extract the DNA and use a technique called ultrasonification, where you have a cesium chloride gradient in your tube, and you spin it really, really fast to create this density gradient. And then you can then fractionate it to separate out your labeled and unlabeled DNA. So a pretty useful technique, although it can be a little expensive depending on your isotope and the number of samples. But when coupled with metagenomics, you can really understand the active microbiome that you're interested in. Of course, you can also take the multi-omics approach, where you couple your metagenomics with, for example, metatranscriptomics. So metatranscriptomics is when we're going to isolate the RNA from the sample, and this can tell us what genes are being expressed, and you can map these to your metagenomes or to your mags that you've assembled from your metagenomes. And this can tell us more about that active fraction. However, again, there are some caveats. RNA is not super stable in soil, so you're really just getting a quick snapshot of what's being expressed. And then there can also be post-translational modification, which you have to consider as well. There's also metaproteomics, which is focused on the peptides that you can isolate from your soil, and metabolomics, which is focused on all the different metabolites that can be isolated. So this could be lipids, carbohydrates, proteins. And these two techniques here, I think, are going to really grow in our field in soils over the next 10 years. EMSL is developing some great technologies to look at this in these complex ecosystems. Right now, one of the main challenges is that they're very database driven with regards to identification. And it also can be rather challenging, again, to isolate it from the soil because soil just wants to hold on to everything. So now that I've kind of given a high level overview, and I know that some of the other speakers will go into a lot more detail about some of these factors, I want to talk to you about how I've applied metagenomics to my studies. The first one is just focused on metagenomes. And the second study, I actually coupled this with stable isotope probing to get at that active fraction. So I'm going to talk to you about how the influence of Amazon deforestation on the soil microbial community composition in this first part. So first off, why did I study tropical ecosystems? Well, they're extremely important carbon sinks. You can see in this infographic that it's showing you the carbon stored per ton or tons of carbon per hectare. And if you look over at the tropical rainforest here, you can see that actually it's one of the only ecosystems where it's about equal. The amount of carbon stored in above ground biomass is about the same as what we estimate in our soils as well. And I think a lot of times when people think of the rainforest, they only think of all the plants, but there's a lot of carbon being stored in the soil as well. However, unfortunately, it's really under attack by humans. You can look at this map here where we're looking at the annual net change of forests between 1990 and 2015. And you can see that along the equator in these more warm tones of red and orange, this is where we're losing our forests. And Brazil here is the highest rate of deforestation, and that's where a majority of the Amazon is housed. And this switches these tropical rainforests from a really strong and important carbon sink to a source. In the Amazon rainforest, specifically in Brazil, which is what I focused on, I found that the main cause is cattle ranching. So they slash and burn the rainforest to make these cattle pastures because cattle and beef is one of the main exports and drivers of Brazilian economy. And so they just slash and burn it, creating these massive homogeneous cattle pastures. So the overall objective for this first part was to focus on how does the soil microbial genomic diversity and functional potential change due to land use change in the Brazilian Amazon. To look at this, I had soil samples from one site in the state of Rondônia down here, marked by a star. And this is one of the main forefronts of deforestation in Brazil. And we took soil cores from the primary rainforest here. So when I say a primary rainforest, I mean that as far as we know from land history, there's been no logging or deforestation on record. And then adjacent to that, we take from a cattle pasture, which has been established with, usually they use this Uricloa brasantha grass. So for my first objective, I focused on how does rainforest to pasture conversion affect just the total soil microbial community composition. And so to do this, like I said, I used metagenomics. And what I found was that deforestation significantly alters community composition. This is a super simple bar graph where I'm showing the relative abundance. But when I did analysis of the change in abundance of these different phyla, we found that 13 out of 34 phyla that were annotated significantly changed. And two really stuck out to me in the data. One was Tomarchiota here, which you can see at this smaller gray bar here, which was almost exclusively found in our primary rainforests. And then Firmicutes, we saw a large expansion and increased abundance in the pasture. Since methane is such a potent greenhouse gas, we also wanted to investigate a bit further into how specific known methane cycling taxa shifted with deforestation. So what you're seeing on the left here is methanogens that's changed abundance in the between our different land use types. And on the right are methanotrophs. So methanogens are ones that produce methane. Methanotrophs are consuming methane. And what we found was that methanopyrus and methanobrevibacter both were in higher abundance in our rainforest compared to methanocella and methanosarcina were higher in our pasture. And one of the differences between these groups, at least what's been described, is that methanopyrus and methanobrevibacter are known to be hydrogenotrophic, meaning that they use hydrogen and CO2 to produce methane, whereas methanosarcina, a lot of them are known to use acetate as their main source of producing methane. And methanocella, although they're hydrogenotrophic, they actually require acetate for growth. On the right, like I said, we're looking at methanotrophs, so they're actually oxidizing methane. And what you're seeing here is methylibium is a facultative methanotroph, which was found in our pastures. So that means it doesn't require, or it's not obligately oxidizing methane, but it can. And methylobacter, methylocella, methylcinus were all in higher abundance in the forest. So if you were looking at this, you would say, oh, okay, so generally speaking, we're seeing an increase in methane emissions from the pasture because these other methanotrophs that are going to oxidize and consume our methane are decreasing in abundance in our pasture. But hold that thought until the next section. For objective 2, I focused on how this conversion between rainforest and pasture affected functional potential. So these are those gene annotations to my metagenome assembly. And although there's a lot of different differences that we found, and feel free to go read the paper here, but one of the main things that I noticed was that there was increased carbohydrate metabolism in our pasture, and that not only was it increased, but there was diversification of the type of carbohydrate metabolism occurring. So on the right, I highlighted a few of the changes that I thought were interesting. One, in the rainforest, I found that some genes associated with lignin metabolism were higher in the rainforest, whereas polysaccharide metabolism and fermentation all were higher abundance in the pasture. And then again, just connecting back to the methane focus of this work, I found that methanogenesis was higher in our pasture, which makes sense given that we had a higher abundance of some specific methanogens and that we know these pastures turn into a methane source. And then the particulate methane monooxygenase, which is a key functional marker for our methanotrophs, so those that oxidize methane, was higher in our rainforest. So again, this matches up with sort of the expectation and what we're seeing with the compositional data. Now, for objective 3, I wanted to kind of hone in a bit more, get a higher resolution look, and focus in on how the genomic diversity and functional potential changes with land use change at the population genome level. So this is using those metagenome assembled genomes. And so from my 10 metagenomes that I used from the pasture and rainforest, I was able to isolate a large number of metagenome assembled genomes that were considered medium to high quality based on the standards set by the MAG, I forget what it stands for. And they represented 10 different phyla, which was nice. We found that there was kind of a range of candidate phyla. So at the time, these were really unknown groups, microgenomates, parcobacteria, rocubacteria, and then also some of the dominant phyla that we think of when we think of soil, the acidobacteria and protobacteria, and then some other groups such as the chloroflexi and TM6. And basically what you're looking at here is just, we were able to look at the absolute abundance of my metagenome assembled genomes across the different samples. And that's great for trying to understand what role they might be playing in the ecosystem. One of the other benefits, though, of getting these mags is that you're able to really dig deep into the metabolism of these unknown microbes or currently uncultivated. And so one that I focused on was rocubacteria, because at the time that I was doing this research, there was really nothing known about this group. And what I found was it has a really diverse metabolism. It's able to actually oxidize methane. This was the first one that found particulate methane monooxygenase gene in the MAG. It's also able to, or at least potentially able to oxidize other hydrocarbons. And it has a number of different pathways that I'm not going to go into detail, but feel free to go and look into this paper a bit more if you're interested. So to summarize, I found that in my pastures, there was an increase in firmicutes, which sort of makes sense. There was also diversification of different carbon sources. We saw that the acetoclastic methanogens and methanogenesis genes associated more with pasture, and that the only known potential methanotroph was facultative. In the primary rainforest, however, we saw an increase in tomarchiota and hydrogenotrophic methanogens. Again, sort of makes sense. These primary rainforests are rather oligotrophic, unlike the pastures. So if you're going to be a methanogen there, you're probably going to be hydrogenotrophic. We also saw that lignin degradation was higher or had the potential to be higher in our primary rainforests, along with methanotrophy and the obligate methanotrophs. And then that Rhocobacteria mag was the first found to potentially oxidize methane and larger hydrocarbons. So this was really great because it kind of gave us this foundational understanding of what could be going on in this land use change. And I wanted to dig deeper, which led to my next talking point, which is about combining soil metagenomics with active microbiome techniques. And I applied this specifically to methane cycling. So the overall objective was, how does land use change impact the active soil methane cycling microbial community in the Brazilian Amazon? And before I dig too deep into this, I wanted to make sure I made it clear that I'm really focusing on the methanogenesis, so either hydrogenotrophic, where, like I said before, they use hydrogen and CO2 to produce methane, or acetoclastic, where they're using acetate to produce methane. And this is an anaerobic process. Methanotrophy, although we know that there is anaerobic oxidation of methane, I only focused on the aerobic form. I did look into the active fraction from sections of the soil core that were anaerobic and didn't see any activity from anaerobic oxidizing methanotrophs, but just so you know, I did look. And so for methanotrophy, we're going to oxidize methane to CO2. So for this study, we actually had two different sampling locations. They sort of represent the main fronts of deforestation in the Brazilian Amazon. This more northern site in the Tapajós National Forest in the state of Pará is along the Amazon River, which is one of the main newer frontiers of deforestation. And then Fazenda Nova Vida in the state of Rondônia is the same general area that previous study was done. And from each of these sites, I took two rainforests, soil coats from two rainforests, the cattle pasture that was adjacent, and then a secondary rainforest. So a secondary rainforest we define as it is known to have previously been a cattle pasture that was then abandoned and it naturally regrew a forest. So there was no human intervention to cause it to regrow, it was all natural. So to give you an overview of my experimental design, I went in and I took soil cores, and we actually kept these soil cores intact for this study. The reason for this is we didn't want to homogenize the soil and to potentially alter the activity levels of these different groups. We wanted to keep it as close to in the field conditions as possible. So we took the intact soil cores and we put them in gas type jars that then we monitored methane over time while injecting with the stable isotopes of CO2, acetate, and methane, along with, of course, the 12C controls for all of those. And after the incubation period of time where we knew that there would be enough incorporation to get labeled DNA, we moved on to the next step where we extracted the DNA. We then did that ultracentrifugation to separate out the heavy and light, and we fractionated to then do metagenomics, where we were able to analyze differences in the community composition and the gene abundance. So First, I want to say that the methane flux was different. So the nice thing about this is that we had in-field fluxes to match up with the analysis that I was doing with my labeled metagenomes. So this is from a colleague of mine who worked on the same study sites, Kyle Meyer, where he published showing that the pastures in Rondônia and in Pará both had significantly higher abundance of methane flux compared to the primary and secondary rainforests. So that's good, and it's nice to have that in-field functional measurement as well. So I wanted to focus on the active microbiome, but we also wanted to understand how that related to the total community. So we did qPCR of the MCRA gene for methanogens. This is a functional gene marker, and found that there was increased abundance in Rondônia and Tapajós compared in the pasture and secondary rainforest compared to the primary rainforest. So that matches up with our expectation that minimally, at least in the pasture, we would expect there to be more methanogens, and that's what we had previously seen in the original metagenomes, right? For methanotrophs, we were a little bit surprised because there was no significant difference in the functional marker for methanotrophy, one of which, this one here is PMOA, but we also looked at the soluble form and saw that, yeah, there was no difference between our primary rainforest and pasture, and the secondary rainforest did have the highest abundance. So that was sort of surprising, but I'll get into the implications of that a little bit later. So for the first objective, I wanted to understand who was actively involved in methane cycling across these different land use types in the Brazilian Amazon. So what I'm showing on the right is just an NMDS plot between the different sites, showing our pastures are in orange, green is our primary rainforest, blue is the secondary rainforest, and then we have our two different sites. And basically what we found is, rather than grouping by the type of substrate that we added or the land use type, it really was most significantly shifted and the most variance explained by the original location. So these locations that I showed you on that map are very far apart, so it shouldn't be, I guess, that surprising, but it is somewhat supportive that our incubation in the lab with those different soil cores didn't significantly shift the community structure. And then the other key takeaway is that all of these orange dots, which are the pasture samples, are more similar to one another than the primary rainforest. There's quite a lot of spread. And this supports findings from one of the colleagues and PIs on this project, Jorge Rodriguez at UC Davis, who found that conversion from a primary rainforest to a pasture causes biotic homogenization. So that was nice to sort of see confirmation we're still seeing that. When we look into the diversity and how this shifts, we found that there was no difference in methanotroph richness between land use types. So to orient you with this graph, on the y-axis, we're having the richness of active methane-cycling taxa. And then on the X are the different land use types. We have primary rainforest in green, pasture in orange, and secondary rainforest in blue. And then the top row here is methane. So this is meaning we're focused on the methanotrophs. And then the middle one is 13C CO2, and the bottom is 13C sodium acetate. So this top one, there's no difference in either site between land use types for methanotroph richness. And for methanogen richness, there was only a significant difference in the Rondônia sodium acetate. Although you can see that for all of these, even if it wasn't significant, the pasture did have the highest richness of active methanogens. So then looking at specific microorganisms that are associated with the methane cycle, found that in the Tapajós in Pará, we found that all, whether it was methanogens or methanotrophs, were actually in higher abundance in the pasture compared to primary rainforest and secondary rainforest, which was surprising. And in Rondônia, all the methanogens were significantly higher in the pasture, and the methanotrophs were in highest abundance in our secondary rainforest. So This is interesting because we expected, based on the previous metagenome results, to see that our methanotrophs were in highest abundance in either a primary or secondary rainforest. So it was surprising to see, at least in Para, that all of them were in highest abundance in the pasture. So how does functional potential change? We talk about just community composition. How do the genes associated with methane cycling shift between these different land use types? What we found was that really none of the methanotrophy genes were significantly changing, at least not as we expected. The only one that did change was between Rondônia pasture, which is sort of this orange color, was significantly higher than Rondônia's primary rainforests, although secondary rainforests did have the highest abundance of the particulate methane monooxygenase. But really, none of these were significant. So this is, again, not really supporting what we expected, which was that the reason we're seeing this higher methane flux is due to decreased methanotroph activity. It appears to be actually driven by increased active methanogenesis, which is what I'm showing here, where we can see on the y-axis all the different samples. They are labeled, the color labels the land use types. So again, the orange is our pasture, green is primary, and blue is secondary rainforest. And then the symbols are whether it was incubated with 13CO2 or sodium acetate. And what we found was that there was a significant increase in methanogenesis genes in Para's 13CO2 incubation and Rondônia's 13 sodium acetate. So this is sort of implying that, generally speaking, Para seems to be driven more by the hydrogenotrophic methanogens, whereas Rondônia was driven more by the acetoclastic methanogens. And overall, active methanogenesis in Para, was driven by the hydrogenotrophic. So between our two very far apart sites, there seems to be differences in what methanogens are driving that methane flux. However, something to note, which I know the group, Klaus and George and some of the PIs hope to follow up on, is that with this, we did have different sampling seasons. So one was the dry season, one was the wet season. So I think the plan is to follow up on this with looking at that seasonality and understanding maybe it's driven by the moisture content, which methanogens are dominating. So to summarize, we found that in the primary rainforest, there was a decrease in our methane production. So it was a methane sink. And generally speaking, we didn't see an increase in methanotrophy or methanogenesis. In general, it seems to be sort of a not driving the methane flux. In the pasture, we're seeing that methane flux really driven by the increased activity of methanogenesis. Also, there's that increased carbohydrate metabolism that I talked about in the first section, and copiotrophic microbes. This could be due, this increased carbohydrate metabolism could be what's sort of feeding that active methanogens. There's more fermentation occurring. The nice thing, though, is that we did see recovery of the methane sink in the secondary rainforest, which we weren't sure if that was going to happen, right? And so that was really positive to see from the land management side of things, where if you do allow a pasture to be abandoned, it can recover its original methane sink. However, future studies really need to investigate further whether we can do this via afforestation, where humans are actively trying to reforest the land, and how long it takes. The secondary rainforests that we looked at were pretty old. They were between 40 and 50 years old. So hopefully it doesn't take that long to allow it to regain its methane sink. So these findings are actually really important when it comes to pasture land management. All the previous research that had used the metagenomics only, not focused on the active methanogens, had thought that the reason we're having higher methane flux is due to that decreased methanotrophy. And if that's the case, there's different strategies we would take to try to increase their activity. But what I found was that it's actually due to an increased methanogenesis activity, so increased methanogen abundance and activity. And so some of the methods that we could use to try to decrease that methane flux is we could try to decrease the pH. So when they do this slash and burn of the rainforest and create a cattle pasture, they also lime the soil, which increases that acidic pH to a more neutral. And in the literature, there's support that methanogen activity does decrease at those more acidic pHs. Another opportunity could be to try to increase the plant diversity, especially having more of these larger trees. So you can see this pasture land photo I have behind you, or behind my text, is that it's really a homogeneous grass. This is actually probably one where there's even a little more diversity than usual. And so if we established more of these trees and less deep root systems, we might be able to decrease methanogenesis activity by decreasing the fermentation that's occurring. Another opportunity could be to look into genetically engineering or naturally engineering Eurocloa species to change how they exudate root exudates, changing the composition to maybe inhibit methanogenesis or to alter the community structure to not support such high methanogenic activity. And then there are known methanogenesis inhibitors that we could test, although that would be potentially challenging to scale up to a field size scaling. So I think with that, I just want to move on to just a few key takeaways that I hope you'll consider if you're going to use metagenomics to study your soils. One is, I hope that I've convinced you that this is a powerful tool to investigate microbial composition and functional potential. It's especially powerful if you're able to combine it with active microbiome analysis techniques, where you're really able to focus in on how the soil microbiome is responding, how it's actively metabolizing substrates of interest. So I think it could be really powerful in your research. With your experimental design, I think hopefully some of the other speakers will touch on this in more depth, but it's important to think about replicates and your statistical analyses. I ran into issues sometimes where it's like, oh, well, I need more samples, but you have that cost benefit analysis of metagenomes are not cheap, although they've gotten cheaper. And you need to consider what your data really can tell you and what it cannot tell you. So with that, I just want to say that we all have a plan and sometimes that plan can go awry, but hopefully I've given you some pointers to take away and to consider while you plan to have less of these challenges along your way. So with that, I want to thank the teams. This was from my PhD work a few years back, and it took a really large team of people to investigate these sites, both Brazilian colleagues and colleagues in the US. And of course, my PhD lab, Klaus Nusslein at UMass Amherst, and all the MAG work. I was trained by Murat Aaron and Tom Delmont, who are absolutely amazing scientists. So thank you all so much. And thank you again for inviting me to speak today.
Thanks, Marie. That was a great talk. Fantastic. I know that there are some questions. There's one in the chat. I'll take that one first. The question is, can you please explain the higher diversification of carbon sources on the pastures? What would that be? Is it cattle related?
Yeah, so I actually think, although we haven't done the metabolomics to confirm this, but I think the thought actually is that the higher carbohydrate metabolism is really due to the shift in the plant diversity. So the Eurocloa species have these insane root systems and a ton of root exudates that are causing a shift in the composition to diversify, although homogenized, but diversify the microbiome, which allows sort of more microbial metabolites and more plant metabolites from those root exudates.
Okay, and we have some questions that came through on the Discord server. There are a couple about the BondCat process. So one was, in order to use BondCat, don't you have to extract the bacteria, but also isolate each microbe in order to determine which microbes are living and which aren't? Or are you just looking at the whole fraction of cells that's living or not?
Yeah. So for BondCat, the way it would work is you would label your soil with that amino acid analog right? Along with in a separate kind of matching replicate, the amino acid analog with a DNA stain. And you do need to isolate the cells, which I think, some people have had success using like nicodens to isolate cells, to then do cell sorting. So you do sort the cells based on that fluorescent signal. And you can then do metagenomics. So you don't have to do single cell genome sequencing. You could pool those active cells and then do metagenomics on it.
And also, is it possible to apply BondCat after the fact? I.e., if I have samples that have already been sequenced and are sitting at -80, can I still use BondCat to get an idea of what the active fraction is?
Probably not, because of the freezing. Most likely when you go through that freeze-thaw cycle, you're going to have cells lyse. You might have some population that survives that process, but unfortunately, it is something that you sort of have to plan ahead for.
Okay, and here's one. This may not be your area of expertise, but do you have an opinion? Which one is better, BondCat or stable isotope probing for metagenomics analysis of sludge samples?
Is Roland on here? I think sludge, I guess it depends. So I think stable isotope probing can be really great if you know what you want to look for. So if you're looking to just sort of look at the whole active community, you could maybe do stable isotope probing where you use the 18O enriched water to just kind of look at the whole community. And Jennifer Petridge's lab has done a ton of work in that. And then if you want to focus in though on something really specific, like I did, where it's focused on methane cycling, I think stable isotope probing is a better route to go than BondCat, because again, BondCat is going to look at the more whole active fraction, unless you do an incubation where you also add a substrate. So it just depends.
Okay, and there were some questions about methodology. So what size soil cores did you collect for the incubation study?
Sure. They were 10 centimeter deep by 5 centimeter across.
Okay. And then some questions here about the taxonomic analysis. What was your strategy to assign taxonomy in your metagenomes? Was it using reads or mags? And also, what was the other one here? For clustering the OTUs, which database did you use and what percentage of OTUs were unknown when you did your classification?
Okay. So for the first part, the taxonomy classification was using the reads, not the assembled. And for mags, I didn't do mags really on the second part, focus on the active fraction. I started to do it, but then I graduated, and moved on with my life. Sorry, what was the OTU database?
Yeah.
Honestly, I'm sorry, I don't remember. I'd have to go back and look at the paper, because that's something that just is constantly changing. What's the standard in the field at the time, and this was done a number of years ago, so I'm not sure what I used at the time. But all of this is published, so please feel free to go and read the papers.
Okay, and then there's a question, did you only focus on bacteria in your objectives, or is it possible to also look at fungi?
Yeah. So most of it was focused on bacteria and archaea. I did, in the methane focused work, that second portion, I did look to see if there was any known methane producing fungi, because I had someone in my department is a mycologist, and he was like, did you know that some fungi produce methane? And so I was like, okay, I'll do my due diligence and go check. And I didn't see anything.
And here's a question of, are the databases robust enough to assemble mags of fungi, or just analyze specific genes such as KZ?
Yeah. So I should defer to others, but from my understanding, the main issue with assembling fungi is just from the metagenomes, is just how large their genomes are. But I will defer to the computational biologists for that.
That's very true.
Let's see here. Trying to make sure I covered all of these questions. Oh, how was the relative abundance of functional traits determined from mags? Was this based on depth of coverage?
How is the relative abundance of traits? I think they're referring to the abundance of the mags across the samples. Is that, am I understanding that correctly? That heat map that I showed?
Possibly, not 100% sure.
Okay, so for that, it was based on the mapping to the samples, and basically it's the coverage that you get from that process. With regards to the, if they're talking about functional annotations to the mags, I don't think I showed anything about the abundance of the genes, so I'm not sure.
Here's a practical question. If we can only budget for Illumina MiSeq sequencing using specific genes rather than metagenomic sequencing, which techniques are still applicable? Would you suggest any other techniques or approaches if a researcher is limited to Illumina MiSeq sequencing?
Yeah. So I think what I would suggest, if possible, obviously we all have to consider budgets, and some of the techniques I've talked about are not cheap by any means. I think if you have the ability to pay a little bit of money to get a stable isotope, then if you could, and you have like a functional gene of interest, combining those two techniques can actually be quite powerful. So for example, I actually did do amplicon sequencing of my methane focused SIP analysis before I did the metagenomes, just because I wanted to confirm that it worked and that it was labeled well enough. And I think that I could have gotten away with just doing that, focused on the MCRA and the PMOA. So I think that's a possibility, or even doing the labeling, but just doing 16S if you don't have a functional marker. So that's probably what I suggest if you could. If not, we all have to just do the best we can. So, you have to work within the, realm that you're able to, and everyone should understand that.
I have one more question from the chat. They asked, how much of the core sample did you use for the DNA extraction?
Yeah, so I didn't go into the details of this part of the method just because it was going to take a bit of time to explain. But the way that I did it was, as I said, we had these cores in the jars, and I was monitoring methane flux every week, checking the activity level. And so because I kept them intact, what I ended up doing was actually looking at, I split them into 5 fractions. So that 10-centimeter core was into 2-centimeter fractions. And I used qPCR to identify the fraction with the highest abundance of that specific functional marker. And then I went through the process of isolating enough DNA to do the ultracentrifugation and everything, and basically confirming that it looked like we had enough label.
Okay, why don't we end the questions there?